Cell growth, specialization, and replication are all regulated by a process called the cell cycle. Mitosis, which is a major part of the cell cycle, is how cells replicate. Before mitosis can occur, the cell is in interphase, which is the longest stage of the cell cycle. G1 of interphase is when the decision to begin this cell division is made. In the S phase, each chromosome in the nucleus doubles and has two identical sister chromatids that are joined by centromeres. In G2, the cell prepares to enter mitosis. After the cell completes mitosis, it undergoes cytokinesis and two identical cells are produced.
Mitosis is important for cell replication and creates two genetically identical daughter cells. In interphase, DNA is jumbled in the nucleus in the form of chromatin. The nucleus is membrane-bound by the nuclear envelope, and there are centrosomes with two centriole pairs.
The cell enters prophase when the chromatin condenses into chromosomes. The centrioles migrate to opposite poles and the spindle fibers form, and chromosomes move towards the center of the cell.
In prometaphase, the nuclear membrane breaks down. Each of the two chromatids of a chromosome has a kinetochore, and some of the kinetochore is attached to microtubules. A non-kinetochore microtubule is not attached to a kinetochore.
Next, in metaphase, the spindles formed and chromosomes take position along the metaphase plate in the center of the cell. Each double-stranded chromosome is attached to a microtubule at its centromere.
In anaphase, the centromeres of each chromosome separate, and the amount of chromosomes in the cell is doubled as a result. The microtubules pull on the chromosomes towards their respective poles, and the non-kinetochore microtubules push against each other to separate the cells.
In telophase and cytokinesis, the chromosomes gather at the poles of each cell and begin to form new nuclei. The nuclear membrane is reformed and the chromosomes unwind. In cytokinesis, the cytoplasm divides and the cells officially separate into two identical diploid daughter cells.
Meiosis is important for reproduction and genetic variation. Interphase of meiosis is very similar to interphase of mitosis in that chromosomes duplicate and prepare the cell for division.
In prophase 1 of meiosis, homologous chromosomes pair and exchange segments to form a tetrad. Non-sister chromatids might cross over at points called the chiasmata. This whole process is called synapsis.
In metaphase 1, tetrads line up along the metaphase plate, kinetochore microtubules attached to their respective kinetochores.
In anaphase 1, the pairs of homologous chromosomes with exchanged pairs separate when the kinetochore microtubules begin to shorten and the whole chromosomes are pulled to their opposite poles.
Telophase 1 in cytokinesis causes cells to split and form two haploid cells that still have double chromosomes. The chromosomes uncoil and the nuclear membrane reforms.
In prophase 2, the sister chromatids reform and the nuclear envelope breaks up as the centrioles move to opposite sides of the cell.
In metaphase 2, the sister chromatids line up along the metaphase plate and the kinetochores attached to the spindle fibers.
In anaphase 2, the sister chromatids separate and become sister chromosomes as they move towards opposite poles.
In telophase 2 in cytokinesis, 4 total genetically unique haploid daughter cells are formed when the cells cleave from each other, the nuclear envelope reforms, and chromosomes unwind.
This table summarizes the major differences between mitosis and meiosis. In prophase 1 of meiosis, synapsis and crossing over occurs, while it does not occur in mitosis. The centromeres split in anaphase of mitosis and in anaphase 2 of meiosis, so single chromosomes are pulled to poles in mitosis, while double chromosomes are pulled to poles in anaphase 1, and single chromosomes are pulled to poles in anaphase 2 of meiosis. In mitosis, there's one cell division and two identical cells result, and there are two divisions that result in 4 unique cells in meiosis. Mitosis, since its cell replication, produces diploid cells, while meiosis produces haploid sex cells.